Ansuvimab. Ansuvimab, sold under the brand name Ebanga, is a monoclonal antibody medication for the treatment of Zaire Ebolavirus infection. The most common symptoms include fever, tachycardia, fast heart rate, diarrhea, vomiting, hypotension, low blood pressure, tachypnea, fast breathing, and chills. However, Ansuvimab was approved for medical use in the United States in December 2020. Chemistry. The drug is composed of a single monoclonal antibody MAb and was initially isolated from immortalized B cells that were obtained from a survivor of the 1995 outbreak of Ebolavirus disease in Kikwit, Democratic Republic of Congo. In work supported by the United States National Institutes of Health and the Defense Advanced Projects Agency, the heavy and light chain sequences of Ansuvimab MAb was cloned into CHO cell lines, and initial production runs were produced by Cook Pharmica DBA Catalent under contract of MedImmune. Mechanism of action. Neutralization. Ansuvimab is a monoclonal antibody therapy that is infused intravenously into patients with Ebolavirus disease. Ansuvimab is a neutralizing antibody, meaning it binds to a protein on the surface of Ebolavirus that is required to infect cells. Specifically, Ansuvimab neutralizes infection by binding to a region of the Ebolavirus envelope glycoprotein that, in the absence of Ansuvimab, would interact with viruses' cell receptor protein, Niemann Pick cone and P cone. This competition by Ansuvimab prevents Ebolavirus from binding to NP cone and neutralizes the virus's ability to infect the targeted cell. Effector function. Antibodies have antigen-binding fragment Fab regions and constant fragment Fc regions. The neutralization of virus infection occurs when the Fab regions of antibodies binds to virus antigens in a manner that blocks infection. Antibodies are also able to kill virus particles directly and slash or kill infected cells using antibody-mediated effector functions such as opsonization, complement-dependent cytotoxicity, antibody-dependent cell-mediated cytotoxicity, and antibody-dependent phagocytosis. These effector functions are contained in the Fc region of antibodies, but is also dependent on binding of the Fab region to antigen. Effector functions also require the use of complement proteins in serum or Fc receptor on cell membranes. Ansuvimab has been found to be capable of killing cells by antibody-dependent cell-mediated cytotoxicity. Other functional killing tests have not been performed. History. Ansuvimab is a monoclonal antibody that is being evaluated as a treatment for Ebolavirus disease. Its discovery was led by the laboratory of Nancy Sullivan at the United States National Institutes of Health Vaccine Research Center and J.J. Mayam from the Institut National pour la Recherche Biomédicale INEB in the Democratic Republic of Congo, working in collaboration with the Institute for Biomedical Research Bellinzona, Switzerland, and the United States. Ansuvimab was isolated from the blood of a survivor of the 1995 outbreak of Ebolavirus disease in Kikwit, Democratic Republic of Congo, roughly 10 years later. In 2018, a Phase 1 clinical trial of Ansuvimab was conducted by Martin Gaudinsky within the Vaccine Research Center Clinical Trials Program that is led by Julie E. Ledgerwood. Ansuvimab is also being evaluated during the 2018 North Kivu Ebola outbreak. Ansuvimab has also shown success with lowering the mortality rate from 70% to about 34%. In August 2019, Congolese health authorities, the World Health Organization, and the U.S. National Institutes of Health promoted the use of Ansuvimab alongside toltevimab/maftivimab/odsuvimab, a similar regenerant-produced monoclonal antibody treatment, over other treatments yielding higher mortality rates. After ending, the U.S. Food and Drug Administration FDA approved Ansuvimab based primarily on evidence from a clinical trial 1/in CT 3,719,526 of 342 participants with Zaire Ebolavirus infection. The trial enrolled newborn, pediatric and adult participants, including pregnant women. All participants received standard, supportive care for the disease. In addition to the standard care, participants were randomly assigned to receive either a one-time dose of Ansuvimab or one of the three other types of experimental treatments, including one as the control group. The participants and the healthcare providers knew which treatment was being given. The trial was conducted at 4 sites in the Democratic Republic of Congo during an outbreak that began in August 2018. Discovery. A 2016 paper describes the efforts of how Ansuvimab was originally developed as part of research efforts led by Dr. Nancy Sullivan at the United States National Institutes of Health Vaccine Research Center and Dr. This collaborative effort also involved researchers from Institute of Biomedical Research and the United States Army Medical Research Institute of Infectious Diseases. A survivor from the 1995 outbreak of Ebolavirus disease in Kikwit, Democratic Republic of Congo, donated blood to the project that began roughly 10 years after they had recovered. Memory B cells isolated from the survivor's blood were immortalized, cultured, and screened for their ability to produce monoclonal antibodies that reacted with the glycoprotein of Ebolavirus. Ansuvimab was identified from one of these cultures, and the antibody heavy and light chain gene sequences were sequenced from the cells. These sequences were then cloned into recombinant DNA plasmids, and purified antibody protein for initial studies was produced in cells derived from HEK293 cells. Ansuvimab and Ebolavirus combination. In an experiment described in the 2016 paper, rhesus macaques were infected with Ebolavirus and treated with a combination of Ansuvimab and another antibody isolated from the same subject, Ebolavirus. 3 doses of the combination were given once a day starting 1 day after the animals were infected. The control animal died, and the treated animals all survived. Ansuvimab monotherapy. In a second experiment, described in the 2016 paper, rhesus macaques were infected with Ebolavirus and only treated with Ansuvimab. 3 doses of Ansuvimab were given once a day starting 1 day or 5 days after the animals were infected. The control animals died and the treated animals all survived. Unpublished data referred to in a publication of the 2018 Phase I clinical trial results of Ansuvimab reported that a single infusion of Ansuvimab provided full protection of rhesus macaques and was the basis of the dosing used for human studies. Development. Ansuvimab was developed by the Vaccine Research Center with support of the United States National Institutes of Health and the Defense Advanced Projects Agency. The heavy and light chain sequences of Ansuvimab were cloned into CHO cell lines to enable large-scale production of antibody product for use in humans. Human safety testing. In early 2018, a Phase I clinical trial of Ansuvimab's safety, tolerability, and pharmacokinetics was conducted by Dr. Martin Godinsky within the Vaccine Research Center Clinical Trials Program that is led by Dr. Julie E. Ledgerwood. The study was performed in the United States at the NIH Clinical Center and tested single-dose infusions of Ansuvimab infused over 30 minutes. The study showed that Ansuvimab was safe, had minimal side effects, and had a half-life of 24 days. Ridgeback Biotherapeutics. A license for Ansuvimab was obtained by Ridgeback Biotherapeutics in 2018 from the National Institutes of Health National Institute of Allergy and Infectious Diseases. Ansuvimab was given orphan drug status in May 2019 and March 2020. Experimental use in the Democratic Republic of Congo. During the 2018 Equator Province Ebola outbreak, Ansuvimab was requested by the Democratic Republic of Congo DRC Ministry of Public Health. Ansuvimab was approved for compassionate use by the World Health Organization, MURI Ethical Protocol, and at DRC Ethics Board. Ansuvimab was sent along with other therapeutic agents to the outbreak sites. However, The outbreak came to a conclusion before any therapeutic agents were given to patients. Approximately 1 month following the conclusion of the Equator Province outbreak, a distinct outbreak was noted in Kivu in the DRC 2018-20 Kivu Ebola outbreak. Once again, Ansuvimab received approval for compassionate use by Humuri and DRC ethic boards and has been given to many patients under these protocols. In November 2018, the Pamojituland Meshapam Together Save Lives open-label randomized clinical control trial was begun at multiple treatment units testing Ansuvimab, Adaltivimab/Maftivimab/Odsivimab Ren-EB3, and Remdesivir to ZMapp. Despite the difficulty of running a clinical trial in a conflict zone, investigators have enrolled 681 patients towards their goal of 725. An interim analysis by the Data Safety and Monitoring Board DSMB of the first 499 patients found that Ansuvimab and Ren-EB3 were superior to the comparator ZMapp. Overall mortality of patients in the ZMapp and Remdesivir groups were 49% and 53% compared to 34% and 29% for Ansuvimab and Ren-EB3. When looking at patients who arrived early after disease symptoms appeared, survival was 89% for Ansuvimab and 94% for Ren-EB3. While the study was not powered to determine whether there is any difference between Ren-EB3 and Ansuvimab, the survival difference between those two therapies and ZMapp was significant. This led to the DSMB halting the study and PALM investigators dropping the Remdesivir and ZMapp arms from the clinical trial. All patients in the outbreak who elect to participate in the trial will now be given either Ansuvimab or Ren-EB3. In October 2020, the U.S. Food and Drug Administration FDA approved Adaltivimab's